Already?
We are recording.
Awesome. Okay, my clock reads 12 Central. I know many of you are joining us from various time zones around the globe. Welcome to the Tree Fund webinar series. And a happy New Year's to everybody. This is our first Tree Fund webinar of the year. We're excited to have all of you back and for another great year of Tree Fund webinars. We have a great list of speakers, and I'll show you those here in a minute coming up. Monica has been working very hard and putting those together and reaching out to a wonderful group of speakers who will be sharing some of the new and emerging research that is coming out of Tree Fund. As is our usual, my name is Bo Broadbeck. I'm an extension specialist at Auburn University, and I will be serving today as the host and lead moderator. With me today is Monica Otting with Tree Fund, who I collaborate with, and we bring this wonderful webinar to you every year. As many of you know, a lot of this topics that we hear from in the Tree Fund webinar series are research that has been funded by Tree Fund. This is made possible by many of our crown sponsors, and you see the list here on this page, as well as our platinum sponsors, as well as from contributions from people like you and I. It takes everybody contributing a little bit to be able to fund some of this wonderful research that we all end up using in our jobs, in our practices. At Tree Fund, we did an analysis a few years ago, and we found that a good many of the standards and BMPs have had some influence from past Tree Fund research. So I say this just to encourage everybody to contribute to some of the work that Tree Fund does. As I mentioned earlier, the Tree Fund webinar series has a great list of speakers for us today, beginning with the presentation March with John Goodfellow looking at integrated vegetation management. We'll then jump across the pond with Andrew Hirons talking about urban stormwater, and you can see the other topics. And so you'll be getting emails about those. And please note that these webinars are recorded. So if you happen to miss this one, or you wanted to catch up with one of our webinars from last year, these are available at treefund.org. Just go to the webinars tab and you'll see all the past recordings. As I've already mentioned, when you make a donation to Tree Fund, it is tax-deductible donation. So help us, support urban tree research and education programs and being able to provide this content to you free of charge. Monica will be posting in the chat box how you can donate to Tree Fund. And so if you find value in these webinars and the research that you're learning, make a small contribution. CEUs, yes, we do have CEUs for you. There are a couple of changes that I want to bring to your attention. What has not changed is that we submit the CEUs for you on your behalf, both to ISA and SAF. During the registration process, we asked for your name as well as your ISA and SAF credential or certification number. What has changed is ISA has asked us that we make sure that when you enter that certification number that you enter it correctly. Make sure in particular that you have that dash mark in the right place and you're using the right capital letters. They are starting to automate this system, and as such, need to make sure that that we don't have a lot of variation there. So please pay attention to that. If you did not provide your certification number during registration, or maybe you have extra people watching with you, just send an email to Monica at treefundwebinars@gmail.com, and she will make sure those get approved. I would like to introduce our speaker today. Before I do that, let me stop sharing my screen so that she can share her screen. So with us today is Dr. Liliana Franco Lara. She is a Colombian biologist trained in biochemistry and virology. In the last 23 years, she has worked for the Universidad Militar Nueva Granada in Bogotá, Colombia. Her research work has been devoted to the study of diseases associated with phytoplasms in urban trees of Bogotá. This includes the symptom description, molecular characterization of the pathogens, insect vector biology, DNA barcodes for insect vector identification, epidemiology of the disease. Lately, phytoplasms, she has incorporated into her work phytoplasm infecting potato and strawberry. Additionally, she also works with potato viruses. So I would like to welcome with us today Dr. Franco Lara, presenting on her work that was funded by Tree Fund a few years ago. Dr. Lara, thank you very much for being here, and I'll turn the virtual floor over to you.
Thank you, Bert. Thank you, first of all, to Tree Fund for financing me, but also for giving me the opportunity to share my research with this huge group of people who are really interested in urban trees. So I am very happy to be here. And well, I'm going to talk a little bit about our work. This work was made by me, but also with my students and many collaborators. So first of all, I want to talk a little bit about epidemiology. Epidemiology has been a word that is becoming has become very famous, very fashionable since the pandemic began. And usually people don't know what it means. So for this talk, I will refer to epidemiology as the sum of the factors controlling the presence or absence of a disease or a pathogen. And in this case, the pathogen is phytoplasmas. When we speak of epidemiology, the disease triangle jumps to mind. But in the case of phytoplasmas, there is really a square, not a triangle, but a square, in which four variables interact. First of all, susceptible hosts, virulent pathogen, insect vectors in this case, and favorable environment. So what I'm going to show you is how these variables interact to produce a very severe disease that affects the trees in Bogotá and surrounding areas. So first of all, I want to locate Colombia. Colombia is located in the tropical zone. The results I'm going to show you are derived from works that were conducted in Cundinamarca, that is a central department in Colombia, in which Bogotá, the capital city of Colombia, is located. The north part of this city is the urban area of the city, and to the south, we have a rural area. Bogotá is located in the Andes Cordillera at 8,500 feet. So it is very high, and there are no seasons in this area. Okay, so phytoplasmas. Phytoplasmas are bacteria of the class Mollicutes. They lack cell walls. They colonize the plant phloem and insect tissues. They're obligate parasites, that is, they cannot live outside of plants or insects. They are unculturable in vitro, transmitted mainly by hemipteran insect vectors, such as leafhoppers and psyllids. There are no chemical treatments available for them, and they are classified in groups or in the genus Candidatus Phytoplasma, but I won't talk about classification, but because that's really confusing, and there is not so much point in what I'm going to tell you. To the right, we have two plants they are from the same species. They were planted the same day, and they were treated in the same conditions. The one to the left is infected with phytoplasmas, and as you can see, it is very different from the one from the right. It is not being exposed to phytoplasmas. So basically what we see here is a departure from the normal architecture of the plant. So I'm going to show you in some details the symptoms that you can expect when you have a plant that is infected with phytoplasmas. These pictures are showing the usual symptoms that you expect to have in if you have a plant infected with bacteria. So you have bacterial spots or cankers, soft rots, wilts, crongles, and slimes. But these are not the symptoms that can be associated with phytoplasmas. And the other hand, phytoplasmas are unculturable bacteria. It's very, very difficult to culture them. And even it has been done, but it is not reproducible. So the Koch postulates cannot be tested. So we have to rely on other kind of evidence to associate the pathogen with the symptoms, with the disease. As I said before, phytoplasmas in nature are mainly transmitted by insect vectors. They can also be transmitted by vegetative material, rarely by seeds, by graftings, but they are not transmitted by contact or mechanical or by mechanical transmission. So Hemiptera is a very large group of insects that feed on plants. And among the Hemiptera, many species into the Cicadelidae and the Psyllidae families are insect vectors of phytoplasmas. That is, they become infected with the phytoplasma when they feed from the plant. The phytoplasma reproduces inside the insect, and it reaches the salivary glands of the insect. So when it goes to a different plant, let's say a clean plant, and it introduces its saliva, it transmits phytoplasmas. Okay, so I'm going to show you a little bit the symptoms that we expect to find in plants that are infected with phytoplasmas. First of all, we have yellowing. This is yellowing can be caused by many pathogens or many conditions, but it's associated to phytoplasmas. So we have leaves that lose their normal green color. We have witch's brooms, that is proliferations from the axillary shoots from one point, resulting in a structure that has the appearance of a broom. Phyllodes, this is a very beautiful symptom. This is a normal flower. These are strawberry flowers. And these two here have structures similar to leaves that develop from the fruits. So this is a strawberry of the fruit. You can see the little leaves developing from the achenia of the fruit. And in this case, the flower, all the flower, the petals were replaced by leaves. And in this, to the right, we have the liquescent branching. And this is the loss of apical dominance, in which the normal branching patterns of the plant are completely lost. So you can see that when you see the plant, they look really funny. I mean, they look strange, and many times it is really difficult to know what are you dealing with. Virescence is also a very nice symptom. In this case, also it's a strawberry flower, and you can see green parts developing from the petals. These are small leaves, little leaves that remain little all the way through the development of the plant. They never turn their normal size. And they can be very, very characteristic. I mean, I follow these in trees, and they say there for long, very long periods of time, epicormic shootings, that is abnormal shooting that appear in the trunks, like in this case, or in the branches, and as I'm going to show you later. Tufted foliage, that is when the internodes of the branches become very close together. So what you see is that the leaves tend to accumulate in one part of the branch. So that when you see the trees, they have these very funny tufted areas in the crown. So there are many more symptoms, but those are the most important ones. So what kind of evidence can you collect when you're studying phytoplasms if you cannot perform Koch postulates? First of all, symptom observation. You have to go out, look at the plants, and compare what you would expect is the normal. DAPI tests, these are microscopy tests in which you stain the tissues with a special fluorescent dye. Electron microscopy. Probably the most important tool is PCR of the 16S RNA gene of phytoplasmas. As you may know, it is known to do the taxonomy of most of the bacteria. And then what we do is we cut those PCR amplicons with restriction enzymes to generate patterns, and each pattern is particular to a group of phytoplasmas. So we can differentiate groups. We can also do daughter transmission. We can use this ectoparasite that you can see here as little hairs to transmit phytoplasmas from trees to plants like this one, to herbaceous plants like this one, so that we can reproduce the symptoms in those plants, symptoms that are known to be associated to phytoplasmas. And also, well, it helps to work with the phytoplasmas in the laboratory. You can do insect transmission assays. You need to find the insect vector and transmit the disease or the pathogen from, for example, trees to other plants. We do sequencing analysis of not only of the 16S RNA gene, but of other genes. And now there are many sequences, there are several sequences of phytoplasmas, of complete genomes of phytoplasmas, and this also is helping a lot. It is very difficult to do. Phytoplasmas are funny bacteria, but there are about now around 20 sequences of complete genomes, and qPCR to measure the amount of phytoplasmas. And this technique gives us much more sensibility than normal PCR. Okay, so let's see what we found in Cundinamarca. So when I began to work with phytoplasmas many years ago, well, I became interested in phytoplasmas because this tree, Fraxinus udei, is a tree that is very common, used to be the most common tree in Bogotá. It is an introduced tree. It was brought to Colombia, to Bogotá in the 1950s from Washington in the US. And although it is native from Mexico. And these trees became the most common urban trees in Bogotá. They used to be very, very beautiful, green with very big crowns, and they were really big trees. So they were very, very nice. But in the 2000s, at the beginning of 2000, they began to show symptoms like the ones you can see here. I mean, you see the tufted foliage, the branches died, and you had the liquescent branching. And so we started to, with a group of people, we started to study the the cause of this disease, and we found out that those symptoms were very similar to phytoplasma symptoms that had been reported in North America associated with group 7 of phytoplasmas. In North America, group 7 infects Oleaceae and especially in the genus Fraxini and Syringa. But we don't have Fraxinus or Syringa in Colombia. But what I'm going to show you is that from those trees, from the Fraxinus trees, they spread to many other species. I am so sorry with you. I need to do something very, very small. Excuse me 1 second. I am sorry. That was my cat. I am sorry. Okay, so this is how I began to work with phytoplasmas. I had been trained as a virologist, and I found out that the techniques that I was using in virology were useful to study phytoplasmas. So this is a liquid amber tree. It is also an introduced tree, and you see here areas of yellowing, and you see tufted foliage and the elongation of internodes. And to the right, you see trees. I mean, the final thing is that the crowns lose their shape. Liquid ambers are supposed to be conical trees, but this is like they look now. In Bogotá, this is Pitosporum Undulatum, introduced from Australia. You can see here branches, witch's broom, branching the ramification of the tear shape of the tree is really strange. You can see tufted foliage, and you can see epicormic growth. This is Quercus humboldti to the right. This is a native tree, very important in Colombia. It is part of the Andean ecosystems, and it is like one of those species that is really, really important. It has been used as an urban tree. It has been planted maybe 20 years ago or something like that. And it has these symptoms in which you can see deliquescent branching. This is croton. This is also a native species to this area of the world. And in these trees, we see this very strange symptom because it produces like little leaves that are very thick and very hairy, and they cover the surface of the trunk. Acacia melanoxylon to the right, you can see epicormic branches that are growing, and leaves in very strange parts of the tree. And these, you can see the elongation of the internodes, changing completely the shape of the tree. In Magnolia grandiflora and Eugenia neometifolia, you can see the loss of apical dominance. When you look at those trees, you really don't know which is the main branch, the main trunk of the tree, and the leaves have become yellow, and they change their shape, and you can see here a lot of leaves in unexpected areas, unexpected parts of the tree. Populus nigra, with very, very elongated internodes. Sambucus nigra, you can see here tufted foliage, yellowing. And Salix humboldtiana, you can see deliquescent branching and a general change in the crown of these trees. These are the trees in which we have performed molecular tests and microscopic tests and several tests. So we know that they are really infected. But we know that there are more species that are infected. And we know that because the symptoms are really evident. But I mean, I don't know if there's any point in just describing and describing and describing something that we know it is there. So, okay. So here I'm showing some evidence of the presence of phytoplasmas in the trees. This is an RFLP analysis. So this was made by PCR. We amplified a gene that is specific for phytoplasmas, and we cut it with a restriction enzyme, RNase A1, and what we see is different patterns. So the yellow ones, the yellow triangles show us the presence of 16S-R1 group, and the red one 16S-R7. And for example, in these two cases, we have a mixture of both phytoplasmas infecting the same plant, because we can see both patterns in the same sample. So our molecular data show that the trees in Bogotá are infected mainly by these two groups of phytoplasmas of group 16S-R1 and group 16S-R7. And that was also confirmed by electron microscopy. You can see the little arrows showing phytoplasmas are very, very small cells. They lie inside the phloem of the plant. That's where they are obliged to live. This cladogram shows sequences of phytoplasmas from different tree species of Bogotá. And what we're showing here is the date cluster with reference sequences of phytoplasmas of group 16S-R7 and group 16S-R1. So what we're saying here is that our molecular tests, these molecular tests, confirms the RFLP results that I showed you before, and that we've tested in with many sequences and many tests. In 2013, we made a large study at the time, in which we studied the prevalence of the disease in the city. So we studied these 9 species of trees, and the presence of phytoplasmas, or the symptoms, were present in 100% of the Fraxinosudei trees. And the less infected species was Croton SPP, in which about 40% of the plants were infected or were symptomatic. And apart from Quercus walti, we haven't done another prevalence study as large as that one. But we know that symptoms, we have not observed symptom remission. The trees, in fact, the disease has progressed, so they look unhealthier. And now, well, this was almost done almost 10 years ago, 9 years ago, long time. They look really, really awful. They look very sad. And but we don't see any remission of symptoms. This has been reported for other phytoplasmas infecting trees, but it's not the case of Bogotá. But another thing is that apparently the trees don't die. That is an interesting observation, because you would expect that if the trees are infected, they would die. And we have Fraxinos trees that were planted in the 1950s, that's 70 years ago, and they are still alive. They look very affected, they are very diseased, but they remain as, maybe I show a picture. If I can go back a little bit, I can show you. They look like this one here. And they stay like that, years go, pass and pass and pass, and they look very much the same. So this is interesting, because phytoplasmas are very good pathogens. They manipulate plants, and as I'm going to show you, They also manipulate insect vectors. And the fact is that phytoplasmas are not interested in killing their hosts. So they apparently, they keep them alive for very long periods of time, making them a very good source of inoculum for other plant species. In 2017, we made a very large study in Quercus humboldti urban trees. We sampled 238 trees. We also sampled the possible insect vectors. And we found that 80% of the trees were symptomatic and 94% of the trees were positive for phytoplasmas of 16SR1, 16SR7, or both phytoplasmas at the same time. At the time in this work, what we were looking at the time is that there were any particular environmental condition that would affect the disease. Something like temperature or temperature or relative humidity or rain or pollution, but nothing, I mean, we couldn't find any relationship with any of those variables. But the disease is widespread in all the city, in all Bogotá. And we don't know if there are, I mean, we haven't been able to find any modulating aspects in the environment. So for some time we thought that phytoplasmas were a problem of urban trees. But at some point, we began to see these pictures were taken from the campus of the university in which I work. My university has a campus in Bogotá and has a campus in North Bogotá, 17 kilometers north from Bogotá, in a little town called Cajicá. And at the time, some people were doing some research in strawberry varieties, and they called me because they thought that they have these very funny things in their fields. And these are some pictures that I took at the time. You can see a normal flower, strawberry flower, and you can see here, this flower has a second round. The petals became leaves. This is the famous strawberry I showed you before, and flower with virescence, and this is a different, you see the fruit became green. And we demonstrated that these plants were infected with phytoplasmas that had a very similar sequence to the phytoplasmas that we had reported in trees. So the pink dots show phytoplasmas from strawberries, and the green dots phytoplasmas from trees. And again, the group in the cluster with 16SR7 and 16SR1 phytoplasmas, and they look very much the same. The phytoplasmas, if you look them at molecularly, they look very much the same. Some years later, potato growers from the region, from Cundinamarca, began to report symptoms of a new disease in potato. This map here, again, is Cundinamarca. Bogotá is in this area, in the central part of the green. The green areas is the parts in which we conducted our potato survey. Colombia is a very important producer of potatoes, and Cundinamarca is the main potato producer of Colombia. It produces about 40% of the potatoes that we eat. And we grow two different species of potatoes. We grow Solanum pureja and Solanum tuberosum. Both species are infected, and they have these symptoms. The leaves become curled. And the margins of the leaves are purple. They show yellowing. Frequently, the plants are very small. They produce less tubers. And well, those are the main symptoms. Now we've done some recent studies, and we know that this disease has spread not only in Cundinamarca, but in other potato growing departments of Colombia. One problem that we have from the pathological point of view is that there is no way to restrict the movement of infected potatoes from one area to another, because the growers won't follow any rule. So they just move the material from one side to another. And we are beginning to see very, very severe problems in the production in several departments of Colombia. So something as I showed you before, in orange, you can see the sequences from the potatoes of Cundinamarca, and in blue, the sequences from the urban trees of Bogotá. And again, the sequences are very close. They cluster very, very closely. In this tree, I have also sequences belonging to other to phytoplasmas infecting potatoes in other parts of the world, but they don't correlate. Right? Different from phytoplasmas in other parts. Okay, so we also tested the presence of phytoplasmas in wheats because we were looking for alternative hosts. Because we were trying to understand how were the phytoplasmas spreading in the ecosystem. So we tested 34 wheat species, and of them, 11 were positive for phytoplasmas of the 16SR7 group. I'm sorry. Okay. One of the species that we tested at the time was Chancrus clandestinus. This is the grass, the most common grass that grows in this area. Chancrus clandestinus was introduced about 100 years ago to Colombia from Africa, and it was introduced as fodder for cows. Basically, it was introduced to feed cows, and it replaced basically all the native grasses in this area, and also it is now used as an ornamental grass. As I'm going to show you later, it grows everywhere. It is very, very common, and we found that Chancrus clandestinus is infected with phytoplasmas of group 16SR1, 16SR7, and also in mixed infections. So these are some of the wheats that we know can be infected with phytoplasmas in Bogotá. And basically what I'm showing here is that they tend to be asymptomatic. We haven't been able to really find a particular symptom that could be associated with these wheats. This is grass. This is like very long grass, but it looks very normal. I mean, the growth pattern are normal, and also the color. Okay, so what do we know about the phytoplasma that we have in this ecosystem? 16SR1b subgroup B, that is the phytoplasma that is it belongs to the Candidatus Phytoplasma asteris species. Candidatus Phytoplasma asteris is the formal taxonomic name as accepted by the taxonomy committee for bacteria. So these are like the normal taxonomic names. So 16SR1b is more or less synonym with Candidatus Phytoplasma asteris. And this species of phytoplasma has originally been reported infecting corn crops in all America. So it is like a normal disease, a normal pathogen associated with corn in North, Central, and South America. In Cundinamarca, this group is found infecting trees, weeds, grass, potato, and strawberry. So as you can see, it infects plants in a very large number of botanical families. And I wrote down here the subgroups that that is making the very, very fine taxonomy subgroups that we have in the trees in Bogotá. We've also detected new subgroups, like there is new variants of these Candidatus Phytoplasma Assays in Bogotá. The 16S R7A subgroup, as I said before, was firstly reported in North America, infecting Fraxinus and Syringa. It is an important disease in North America. There were many studies many years ago, made in the University of Cornell. And at the time, until the year 2000, it was believed that this pathogen was only found in North America. But in fact, I mean, it came to Colombia, and as I show you, it spread to many botanical families apart from Oleaceae. So in Cundinamarca, it's found in weeds, in grass, in potatoes, and strawberries. And in Bogotá, we have the 16S R7G subgroup that is derived from the 16S R7A subgroup. So what we're saying is that probably in these years, since these phytoplasmas came to Bogotá, they have evolved, and now they are a little bit different from the ones that you would find now in North America. In Liquidambar styraciflua, we've also detected these other groups of phytoplasmas, 5B, 9, and 12. And these phytoplasmas are found in other crops in Colombia. Okay, so for many years, we studied only the phytoplasmas on the trees. But at some point we said, okay, we need to learn something about the insect vectors if we want to control the disease somehow. So I have to say I'm not an insect person. I am not an entomologist. So all this work has been done with the help of entomologists, especially Mike Wilson from the Museum of Natural History of Cardiff in the UK. And of course, my students who sample a lot. And this picture sheet here, this slide here shows us the most common Cicadelidae that we can find in Bogotá. In this study, we sampled during 1 year in 2 spots. One an rural spot and another an urban spot for Cicadelidae in the grass. So we went to Cenchrus clandestinus and we sampled the insects. And we have here the most common ones, pictures of the most common ones. And the 2 most common ones in both locations were Amplicephalus fonsaensis and Exitianus atratus. And I'm going to show you that whenever these 2 species appear, there are 2 pink stars. Okay, these are the Cicadelidae that we've found in Cundinamarca so far. So what I wanted to show, these have been found, this picture is grass and trees. But we know that there are many more species that we haven't detected. As I said before, I am not an entomology person, so the identification of these species has been very difficult. And There are no Cicadelidae experts in Colombia. Most of these species are new to science. They haven't been described. So this is also, this is difficult because we don't know anything about the biology of these insects. We know for some of them that they reproduce in the grass, but that's it. We don't know anything about it. And I'm showing here that part of this work was funded by Tree Fund. Okay, so here we have, we did some years ago, we did some tests trying to learn which of these Cicadelidae insect vectors, leaf hoppers, carried phytoplasmas. When we say that phytoplasmas carry phytoplasmas, we cannot be sure that they are insect vectors. To demonstrate that they are vectors, we have to perform the transmission assays. And transmission assays are difficult to perform. One of the limitations that we have is that we cannot, it's been very, very difficult to raise the insects in artificial ways. So basically what we do when we do our transmission assays is that we pick up the insects from the grass, from infected areas of the grass. We know which areas are infected in the university. So we go there and collect the insects and we make transmission tests. So again, so these here, the ones that I'm showing with a cross, with a, these are positive for phytoplasmas. We know that they can carry phytoplasmas. And we've developed DNA barcodes for their detection. So, and because we have all these limitations to detect phytoplasmas in them, we've developed DNA barcodes for their identification. This slide here shows us the results of transmission assays that we performed with Amplicephalus funzaensis and Nexitianus atratus some years ago, in which we demonstrated that these two species are really vectors. They do transmit. We don't have tested other species so far. In this chart here, what we have is, we compared at some point, this was funded by Tree Fund, we compared the insects that we can collect in Chencrus clandestinus and in Quercus humboldti, that is trees and grass. And amazingly, we didn't expect to find these. We found out that many insects that are in the grass are also visiting trees. Something similar was made for potatoes. So we found out that these species that I'm showing in blue are present in potato and in grass. And the ones that are shown in green are basically the same with the green stars, are also found in the trees. So if we go back to the environment, as I said before, Bogotá has a permanent, constant temperature that is about 68 Fahrenheit during all the year. We have, and in the night, about 42 centigrade, 42 Fahrenheit. And we have dry and rainy seasons. We have 2 rainy seasons per year. We used to have 2 rainy seasons per year, but now that is not like that anymore. And we don't have a winter period. So the phytoplasmas, the infected plants and the insects remain all the year. Grass is an asymptomatic carrier of both phytoplasmas, is present, for example, in Bogotá, I'm showing here near house. And this is where a potato crop. And grass is really interconnecting the rural and urban areas in Cundinamarca. Because grass, as you can see here, well, part of it is some crop, but grass is very, very common. Okay, so what are the conclusions? Phytoplasmas were originally present in corn or in Fraxinus, in 16SR1B in corn and 16SR7 in Fraxinus when it was brought to Colombia. And of course, these areas are also, these plants are also surrounded by grass. So the phytoplasma somehow moved to the urban trees where they are surrounded by grass. So we have grass. And probably from the grass, these phytoplasmas moved to potato crops and strawberry crops in the area. And in all these ecosystems, we've been able to find Exitianus atatus and Amplicephalus funzaensis, the insect vectors of these phytoplasmas. So these are the conclusions. Phytoplasmas of the 16S are, of both groups can infect many botanical families if a proper insect vector is present. Several botanical families are susceptible to the infection by these two phytoplasmas. Tropical conditions favor the dispersal of phytoplasmas because plants and insect vectors are present during all the year. And grass, Cenchrus clandestinus, plays a major role in the pathosystem in Cundinamarca because it is the main host of insect vectors and a susceptible host for phytoplasma. Finally, I want to acknowledge my collaborators, my students, who actually have done all the work, and also the financing bodies, including Tree Fund, who have given me the money to do this. And thank you very much for your attention. And I hope we have some minutes for some questions.
Thank you very much, Dr. Franco Lara. Fantastic presentation. And we do have a number of questions from people. Somebody asked right up at the top, and I know you kind of mentioned the temperatures. Do you know what the hardiness zones are in Bogotá?
The what? Sorry,
the hardiness zones. It's a system we have, I guess, here, I guess mostly in North America, for looking at the average cold temperatures for the year. So
okay. The average cold temperature is probably 0 centigrades.
Okay.
32 Fahrenheit. That's the coolest.
We also had a question that somebody asks, have you found that these phytoplasms affect conifers?
Infect what? Sorry.
Conifers, pines,
yes.
fir,
yes.
Yeah.
They do infect conifers. I think they do because I didn't put them here, but I have these fantastic pictures of conifers, of pines infected. And they look really, really strange. They look very funny, and they are in the middle of the city. So yeah, it is very likely that they do infect conifers.
Thank you. And I had another question here from Scott, and he asks if the infected trees you are showing are intense urban environments. Do you see infections in trees that have adequate soil volume, root flares exposed, inadequate soil moisture? I think he's getting at the idea that there's probably
yes.
stressors involved, the kind of that correlation between those stressors and the phytoplasms.
Yes, I didn't show it here, but Bogotá is located in an area that is called the Sabana de Bogotá. And Sabana de Bogotá is like a large plateau. In the middle is Bogotá, and it's a flat area with agricultural lands. And yes, the trees have symptoms, especially Fraxinus is symptomatic everywhere, but also Quercus, Croton, yeah, they do have symptoms, even if they are in a suitable place.
And related to that, because there was a question here, suggesting that, we don't have chemical control options. And so as you look at management for phytoplasms, how are you addressing management of this pathogen? Is there certain species that are less susceptible that you might encourage planting, or how do you know how that's being addressed in Colombia?
Yes. Well, that's the question that I fear the most, because when I began to do this work, the idea was to try to find some management strategy. But I mean, the management would be to get rid of the grass. That would be the perfect solution, but that is not going to happen. I mean, grass is everywhere. It covers everywhere. It is a very, very competent weed. So yes, for example, croton and quercus tend to be a little bit more resistant, or at least they don't look as bad as the other three species. So, but it is very difficult because you would have to replace the trees, and replacing the trees is going to cost a lot of money. So in this moment, the priority is the money is going somewhere else, as you can imagine. So we're doing nothing about it. Apart from doing some descriptions, we are beginning to make some studies to try to understand if we can use pheromones for these insects to try to avoid them. But in the case of Bogotá, it is going to be, in practice, very, very difficult to manage the disease because it is spread everywhere. So I don't know if someone has an idea. We'll be very happy to discuss it, because I don't really know what we can do.
Absolutely. I mean, that's a challenge because there's a number of questions here related to integrated pest management, trying to natural predators for the cicadas, which seem to be the primary vector for spreading this. But the impracticality of that, I imagine, is certainly a challenge. Let me see here. We've got a minute for another couple questions. Monica, are there any that jump out at you?
Yeah, there's one in the chat box. Someone's asking, Mimi's asking if you have seen this also in cities that are not in such a high altitude, for example, on the coasts.
No, we've seen this. This is very restricted so far to this ecosystem. There are other phytoplasmas found in other places in Colombia and more, yes, but they are associated to other groups of phytoplasmas, mainly phytoplasmas of group 3 and group 5. But group 1 group 1 has been reported in corn in Tolima and Huila that are very, very hot places. And group 1 is known, it is widespread in all the world. It is found everywhere in the world, and it adapts very, very well. It evolves very quickly. So it is a very interesting pathogen. But group 7 so far, we've only detected in Cundinamarca.
Well, we are getting low on time, and I know there's tons of more questions, and I certainly have some more questions, but in the interest of time, I think I'm going to bring us to a conclusion to our webinar. Thank you very much for a fascinating talk. I think you gave us a lot to think about with these, new pests that are out there. And I think, I really appreciate you sharing that research with us. Just in conclusion, to all of you listening in, I know there were some questions about CEU. Those on your behalf directly to ISA. So when you registered, you should have provided your name as well as your CEU certification number. Please note that ISA does require you to have submitted it in the right format. And I think we have that in the registration for this webinar.
Correct.
If you have any questions for that, please email Monica at Tree Fund. Monica, what's that email address? Can you throw that in the chat?
treefundwebinars@gmail.com.
It's easy to remember, treefundwebinars@gmail.com. Any of those questions that we have out there. I really appreciate everybody joining us in. Thank you very much, Dr. Franco Lara, for a wonderful presentation enlightening us on this relatively new disease and new topic to probably a lot of us out here. And we look forward to everybody joining us again in our March webinar, looking at some integrated vegetation topics with John Goodfellow. Just go to treefund.org to be able to see some of that information. Of course, this was recorded. If you wanted to go back and rewatch some sections, because there was lots of material provided today, that should be up on the Tree Fund website, probably later today or sometime tomorrow, depending on when we get all of that stuff put together. Dr. Franco Lara, thank you very much. Monica, of course, thank you always for bringing all of this together for this webinar series. So with that, I will go ahead and close this out as we have reached the top of the hour once again. Thank you.
Thank you all.
Thank you. Thank you. Bye-bye.